So now in bio class, day 10, I'm going to continue in chapter number 4, where it is how cells could obtain the energy, plant or animal cells. So here you will be basically getting the insight about energy and metabolism, glycolysis, citric acid cycle, oxidative phosphorylation, fermentation, and connections to other metabolic pathways. Now, so if you want to understand what is energy and metabolism, this is quite clear. So if you want to obtain energy, you need to go for metabolism of some biomolecules. Here, you get the energy, your cell gets the energy from glucose molecule, and you get the energy in the form of something called ATP. So this is like metabolic pathways or metabolic process. What is metabolic pathways and what is metabolism? These things you need to understand. There are a large number of metabolic pathways. If you want to understand, you can also check some of the pathway databases such as KEGG pathway database or some other pathway databases, but you need to understand what is metabolism, then you need to understand aerobic, anaerobic, anabolic, catabolic. So what is anabolic pathways? The definition for anabolic pathway, and what is the definition for catabolic pathway? So here it is anabolic means something to build, that means to build polymers, catabolic something to break. So here when you are saying breaking something, then definitely you can understand in breaking what it could happen. So the two possible processes is like are anabolic pathways and catabolic pathways. So the first requires energy to build some polymers, you need energy, but to break down, when something is broken down, then the energy will be released. So it is important to know the chemical reactions of metabolic pathways which do or do not take place. Okay, and definitely they do not take place on their own. They need something as a support or initializer or some energy. So enzymes are playing quite important roles for catalyzing all types of biological reactions, and those that require energy as well as those that release energy. So you have third thing in this purpose that is called metabolic enzymes. So anabolic is small molecules are built into large biomolecules, and thus energy is needed. And this is what you are seeing here. Then in catabolism, you break down the large biomolecules into small molecules where you are observing that the energy is released. Now, plant takes energy from sunlight, but animals have different source of energy, which we can see what is for plant and what is for animal. Then another term comes that is called thermodynamics. So which refers to the study of energy and energy transfer, which involves physical matter. Then in thermodynamics, you can try to look for some basic knowledge only, not much. So here you can see there is something chemical energy and then converted to kinetic energy. Light energy is converted to chemical energy. What are the purpose to show here? You need to understand, and then how these cells are using energy. So all the activities of the organism involve chemical reactions. The total of all chemical reactions jointly called metabolism. And cells need enzymes for these chemical reactions to occur or control. Enzymes are special proteins that regulate how fast or chemical reactions should take place at which time also. So enzymes are sometimes functional when they get certain strength. So you can see something called threshold activation energy, then reactant activation energy, then something is formed called product. And the activation energy is somewhere here. But what you need to understand here as a key point is activation energy and at which condition these activation energy is sufficient. So this is quite important. And then another term is coming active site, binding site, substrate, and the products. For example, here you can see substrate, active site, enzyme changes shape slight as substrate binds. After binding, there is structure deformation, and then some products are formed. So what you are seeing here, substrate entering the active site of enzyme. So this is the active site of the enzyme. Then enzyme substrate complex is formed for certain time. Then enzyme and product complex you can see here, and this complex is converted back to product formation and leaving the active site of enzyme. So there are certain models for this purpose to understand this activity, and those are something called competitive innovation, non-competitive innovation, allostatic innovation, and one more term that is called induced fit model. So what is allostatic innovation and what is allostatic activation? So here what you are seeing is in allostatic innovation, it works by indirectly inducing a conformational change to the active site. Now, in with respect to allostatic inhibition, the activation, allostatic activation is the activator molecule modifies the shape of active site to allow a better fit of the substrate. So then what is the role of these enzymes and how this concept is useful for our daily life? Then it comes pharmaceutical drug development process. So, of course, there is a potential role in metabolic activity, but it also helps for the drug developer and how they could do it in pharmaceutical purpose, then or for drug development purpose. It could be like enzyme binding to substrate, similar to what you have seen, then catalytic reaction, then product formation, and then release of the enzyme for the reuse. Now, when you are working deeply into drug development purpose, then you need to investigate the pharmaceutical properties, pharmacokinetic properties, that is also called Swiss admit properties, then pharmacodynamics property, that means the effect. And after that, what you expect as an outcome to evaluate is bioavailability, volume distribution, clearance, and biomarkers. Then clinical outcome, negative, that means it could be possible that the drug which you developed could be worse or toxic. Positive means it improves the effect. or optimal, what you need it for diagnostic purpose. So what you need to understand that in data science, if you are working with a pharma company, you will be getting the data from pharmaceuticals. That means compatibility study, solubility stability study, or pharmacokinetic properties list like absorption, distribution, excretion, metabolism, then pharmacodynamics, that is the final effect of the drug. Then finally, you could also get the data to be analyzed from if you are working for the pharma company, that is bioavailability, volume distribution, clearance, and then clinical outcome. So this is how you need to understand the this basic biology where you can also get a real application in some new emerging field, that is data science. Of course it is not related to data science, this is purely for biology purpose, but still I am discussing now. It comes to feedback inhibition of metabolic pathways. So feedback, from the name itself, it comes that if you are trying to have certain kind of loop, which is basically trying to giving feedback, and inhibition, that means you are trying to block it. And this is what you can see from this figure, that this is one of the finest example of feedback inhibition. But if you want to go for definition, then you can say it involves the use of reaction product to regulate its own product for further production. So this is how you need to control it. So here it is quite important and common in case of biological function, that is biological function of metabolic activity. Then you can understand there are some kind of pathways which you need to know, that is glycolysis. I'm not going to discuss the complete reactions, but what I'm saying, you can see here that from a glucose you can generate some energy, and that energy is in the form of ATP. So here it enters the cytoplasm from the blood, that is glucose. Then two ATPs are used to get energize the glucose, and then glucose splits into, and two 3 carbon molecules are made. Each 3 carbon molecule gives high energy electrons and hydrogen to make NADH, and each 3C molecule then gives energy to make 4 ATP molecules, that is 2 each side. Then each 3C molecule now becomes a molecule of pyruvate. And then finally, the NADH carries energy to the mitochondria to be used for ETC, electron transport chain. And this is how you can understand the glycolysis process. So now it comes a new term, that is ATP, and that is called adenosine triphosphate. So here a living cell cannot store significant amount of free energy. So excess free energy would result in an increase of heat in the cell, which would denature enzymes and the proteins. Thus, could also lead to the destruction of the cell. So a cell must be able to energy safely and release it for the use only at the time of requirement. So living cells do this by using ATP, which can be used to fill any energy need of the cell. So it functions as a rechargeable battery. So when ATP is broken down, usually by the removal of its terminal phosphate, energy is released. And this energy is used to work by the cell, usually by binding of the released phosphate group to another molecule, thus leads to activation. Now, how this ATP could look like? The AC ATP could look like, here you can see triphosphate, three phosphate groups. So this ATP is an organic molecule that acts as the potential energy carrier in cells. And the formula, if you want to write, it could be written as it is mentioned. Then after this, you glycolysis, you go for some summary of the glycolysis. That is, glycolysis is the metabolic degradation of 6 carbon sugar molecule, that is glucose, which splits into 3 carbon molecules that are further processed, leading to produce pyruvates, water, and energy. So what you get from glucose is pyruvates, water plus energy, and that energy finally is to ATP. So there are other such process that is called citric acid cycle and oxidative phosphorylation. So this citric acid cycle, here you can understand that in eukaryotic cell, the pyruvate molecules produced at the end of glycolysis are transported into mitochondria, which are the sites of cellular respiration. If oxygen is available, then aerobic respiration. So here it comes the term of aerobic and anaerobic respiration. So in mitochondria, pyruvate will be transformed into 2 carbon acetyl group by removing a molecule of CO2 that will be picked up by carrier compound called coenzyme A, CoA, which is made from vitamin B5. And the resulting compound is called acetyl coenzyme. So acetyl coenzyme can be used in a variety of ways by the cell, but its major function is to deliver the acetyl group derived from pyruvate to the next pathway in glucose catabolism. Like the converse, so now there are certain steps, as I mentioned for glucose. In the same way, you can also summarize the citric acid cycle and try to understand. Then next part is oxidative phosphorylation, and from the name itself you can see oxidation and phosphorylation, so how it could work. So you need to understand about this oxidative phosphorylation and what are their roles. Then we will be also discussing, other than oxidative phosphorylation, the electron transport chain, then chemio osmosis, osmosis, and then what are the amount of ATP produced. So all these things we will be discussing in the next class. So thank you very much and subscribe this channel for the further update.